Hi, welcome back to Brookings Biology video PowerPoints dealing with macromolecules. In our last PowerPoint, we talked about carbohydrates, one of the important macromolecules for living things. In this PowerPoint, we're going to talk about our next kind of macromolecule, which is proteins. Proteins are made out of carbon and hydrogen and oxygen, just like we saw carbohydrates. But one difference now is we're also going to add another atom which carbs don't have, and that's nitrogen. If you look at this little subunit that we used to make carbohydrates with, notice here's that nitrogen group. It's called an amino group, nitrogen with hydrogens attached, and it's important in one of the important parts of this little building block that we're going to use to build proteins. It's called an amino acid, and that nitrogen group is what gives amino acids their name, amino group. This little building block has a carbon in the center. And remember, carbon is important because it can hold hands with 4 different atoms or groups at the same time. So attached to our center carbon, gotta go back a little bit. Attached to our center carbon, there's a hydrogen. There's that amino group with nitrogen in it, and there's this group. It's a carbon with some oxygens attached to it. Oxygen and hydrogen attached to the oxygen. It's called a carboxyl group. And we're not going to worry about writing out the formula. I'm not going to ask you to be able to write it on your own, but you should be able to recognize it. If you saw a molecule that had these groups attached to it, you should say, oh, I know, that's an amino acid. What makes the 4th attachment to our carbon is this group. It's called the R group. It's kind of like X in math if you've done algebra and used letters to stand for numbers in a chemical or in a math equation. The R kind of stands for something that could hold that place. And so we could add different things into this spot and build different little subunits that we use to hook together to make a protein. If you look at, remember in our last slideshow, we talked about using pop beads as an example of how molecules go together, and we talked about those little toys that kids play with. Each of these little pieces is a monomer piece and can join those pieces together to make a bigger molecule. When we made polysaccharides, the little pieces that we used to join together were glucose molecules, sugar molecules, and I joined them together to make a bigger polysaccharide. We're going to use little pieces again, and we're going to join them together to make a bigger piece, only this time if I'm making proteins, instead of using glucose molecules, I'm going to use amino acid. So amino acid, amino acid, amino acid, amino acid hooked together in a chain, I get a protein. Glucose, 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 I get a polysaccharide. It's the same idea, taking little pieces, monomers, and putting them together to make a bigger piece, a polymer. And proteins are polymers. So we're going to go back to our slideshow, and this is our building block now, our little piece that we're going to use to build and join together to make a protein. So what is different about the different amino acids that I use is that R group. That's what's going to make the little subunits different from each other. So if you look at the basic arrangement of the atoms in an amino acid, you see that center carbon, you see that amino group with nitrogen, you see the carbon carboxy group that's attached here, and hydrogen. And if you look at some examples of different amino acids, you see carbons in the middle, those nitrogen amino groups. They all have the carboxy group and the hydrogen attached. Those are all 3 bonds off of that carbon are the same in every amino acid. What makes them different is what I stick in that R spot. And just like X in math, different things could go into that R spot, different groups, different atoms. So for example, if I attach these atoms, carbon and 3 hydrogens, I get an amino acid called alanine. It's different than this amino acid. This is a different R group. It's got a carbon and some hydrogens, but this one has an oxygen. The R group is a little bit different, and it makes a different amino acid called serine. So if you look at those structure of an amino acid, what you see is they all have the basic part that's the same. These parts here, the carbon in the middle, the amino groups, the carboxyl groups, the hydrogen. What's different is what I put in the R group. The R group is different for each amino acid. It could be something very simple, like just a hydrogen atom. This is the simplest amino acid. It doesn't have very much attached for its R group. But remember, carbon can make some really cool shapes. It can make long chains. It can make rings. And so what I stick on in that R spot is going to be different for different amino acids. You don't have to memorize their structures. You don't have to memorize their names. But you should know the basic overall shape of an amino acid. There are 20 different amino acids that cells use, living things use, to build proteins. So we're going to take these little pieces and we're going to hook them together to make a protein. Just like we hooked glucoses to make a polysaccharide, we're going to join amino acids to make a protein. The cool thing, if you look at other living things besides humans, like bacteria or plants or worms or fungus, they use the same 20 amino acids that we do. There's a connection. Not only do all living things share the same DNA code, universal genetic code, but they also share, they're talking the same protein language. They use the same amino acids to make their proteins. So think about all the possible things that could go into those R spots. It's pretty amazing that living things all use the same 20 amino acids to make their proteins. It shows a connection. It's an example of that connection between organisms. Shared common ancestor, shared biochemistry. Okay, so let's think about how could I take those little subunits and put them together to make a bigger molecule. Remember when we joined carbs, joined sugars to make a polysaccharide? We used a reaction where we removed a water molecule and joined the leftover ends together to make a bond. If you look at these amino acids, here's my nitrogen group, here's my carboxy group on each one, and there's that R group hanging down, kind of like a chain, like a charm bracelet with little charms that hang off of the chain. Those R groups are going to be different in different amino acids. So we have R1, R2, they're telling you that those groups are different. So can you look at this molecule and see where I could get some hydrogens and oxygen to make a water molecule? If I do that, if I take an OH from one side and a hydrogen from the other side, I can pull those out, right, and put them together to make one H. I can remove a water molecule, and the places that are left over now are going to join together to make a bond between the two amino acids. We removed the water. What was our word for that? Remember? Dehydrate means to lose water. And we're going to use that same chemical reaction that we used to make a polysaccharide. We're going to do dehydration synthesis again. Remove a water molecule, make a bond. Dehydration, lose water. Synthesis means to make or put something together. And that same chemical reaction works no matter what little subunit that we're using. We're going to pull out a water molecule. We're going to make a bond to join those little pieces together. If you look at the sequence of the amino acids in a protein, that code, where I put which amino acid in what order, is determined by your gene code. The DNA, when you read a piece of DNA as a gene, is telling the cell to do something. What it's really telling the cell to do is how to make a protein that's going to do something. And so the sequence of amino acids, what goes together, where the R groups go, is determined by your gene code. Once I get the sequence built, though, those molecules, those protein molecules, don't stay in a straight line in the cell. They start to fold up. And remember, we had these vocab words, phobic and philic. Remember, phobic means that I'm afraid of, means fear of water. And molecules that have R groups that are phobic want to try and stay away from water molecules. Remember, phobics are very nonpolar. Molecules, R groups that are hydrophilic, want to touch and be near water. And they are what help to make the 3D shape. What's put in those R positions help the protein fold up into a 3D shape. So think about it. This is a protein floating in a cell. Out on the outside here is water, right? Basically, you're built out of water. Your blood is made out of water. The cell contents of your cells are mainly water. And so think about this molecule. If it's going to fold up, and look at, here's your chain, amino acid, amino acid, amino acid in a big long chain. If it's going to fold into a 3D shape, think about where those R groups want to go. If it's an R group that's polar, that likes water, that's philic, it's going to want to end up here on the outside of our molecule, touching the water, right? It likes to be near water. If you're a phobic R group, one that doesn't like water, wants to stay away from water, if I'm an R group on an amino acid in the chain that's very phobic, I want to be inside here. I want to be inside the molecule. I want to be as far away from water as I can. And so those interactions between the R groups are what's going to make my molecule fold up and get a really cool 3D shape. And remember, shape is going to determine what your job is. How you look, what your shape is, determines what this molecule is going to do for the cell. Once I get my 3D shape built now, remember we have some bonds that we learned about. We talked about van der Waals forces, attractions, tiny attractions that move and change position but help molecules stick together in hydrogen bonds. Once I get in position and get my 3D shape, attractions between different parts of the molecule are going to help to hold that shape and keep it in place and keep it doing its job. Where do we get proteins? They're in foods that come from animals, right? Muscles are proteins. So if we eat an animal muscle from a cow or from a chicken or from a pig or from a fish, we're getting protein. Nuts also have protein and eggs have protein. So let's talk about what are some things that proteins do for us. We already talked about this molecule back in our carb slideshow because this is a combination molecule. It's part protein and it's part carb. The glycol, think glucose. It's glucose. It's a sugar molecule, sugar that's attached to a protein. It's a combination molecule. And the protein part is what sits in the cell membrane, anchors the molecule. And the sugar part, remember, sugar, 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 these little hexagons, the sugars are what sit on the outside surface of the cell, and their job, if you remember from our last slideshow, their job is to help the cell recognize things that don't belong. Are you like me? Recognize myself? Or are you different? And they're important in when we talked about things like blood types, in organ rejection, in recognizing cells like germ cells that are not supposed to be in our body. It's how we recognize and identify things that are different. So glycoproteins are important. One of the functions of proteins, the protein is part of that glycoprotein molecule. Another thing that proteins do is that they can be structural. They're building blocks. Proteins and phospholipids are the main components in building cell membranes. If you look, we talked about phospholipids before, these little molecules with the heads and the tails that hang down. They go together to make a cell membrane, and part of that cell membrane, spaced in between the phospholipids, are proteins. Proteins are important components in building cell membranes. other things that proteins do. They also are structural in that they combine with DNA to wrap into chromosomes. You have a lot of DNA in your cells. Human body cells have 46 chromosomes. If I was to take a chromosome and unravel it, what I'd find inside is a big, long, one piece of DNA. And in order to get it to fit inside my cell and make it so it doesn't get tangled and doesn't get broken, is I need to take that DNA molecule and I wrap it and coil it very tightly to make a chromosome. And the molecules that help that happen are proteins. These little guys here, the little proteins whose job it is just to coil and wrap up the DNA very tightly to make a chromosome, they are proteins. So that's a pretty important function for proteins in your cell. Another thing that proteins can do for you is they act as enzymes. Enzymes are molecules that help chemical reactions happen faster. So when we talk about chemical reactions this year, we talk about breaking down food molecules, we talk about putting molecules together to make carbs or proteins or nucleic acids, all the molecules we're building. Any kind of a chemical reaction that happens in your body, there are enzymes, helper molecules that help those reactions happen. So if you look at this little animation, this is an example of how an enzyme helps you. So this big green molecule here, this big green molecule is the enzyme. And it's going to help this little pink molecule to do its chemical reaction. Remember, some reactions break down molecules into smaller pieces. An enzyme is going to help that happen. So here comes my little pink molecule. It's going to fit right into the enzyme, kind of like a lock with a key. And the enzyme is going to help it do its reaction, and out come the products. Notice the enzyme didn't change. It's ready to go and ready to help the next molecule. So enzymes are reusable. They grab onto a molecule, they help it do its chemical reaction, release the products, and then they go again. And many of the enzymes that work in your body are proteins. Proteins also control genes by turning them on and off. If you remember in our very first slideshow, way back in chapter 1, we talked about how a stem cell, an embryonic I can be anything cell, could turn into a muscle cell or a bone cell or a nerve cell. And we said it happens because those cells are turning on and off different genes. Remember, all cells have the same DNA code. What makes a brain cell different than a muscle cell or a skin cell is which genes those cells are using. Are they turning on a gene? Are they turning off a gene? And proteins are those on-off switches. If you look at this little animation that's going here, this long stretch of DNA, piece of DNA, notice there's genes here. In order to read the information on the gene and use it in the cell, molecules have to be able to get into the cell to read the gene code. Okay? And if they can't do that, if something is blocking the gene, right? If something is blocking the gene, it's going to prevent us from reading the information and using that gene. The little molecules that sit on the front of a gene and block it so we can't use the information, kind of like a roadblock stopping you from going down the road. The molecules that do that, that sit on in front of the gene and turn it off, are proteins. So they're important in controlling genes and how they work. Another thing that proteins do for you is they actually fight the germs. Antibodies are molecules made by your immune system. Once those glycoproteins recognize something is a germ and it's not supposed to be in your body, your immune system sends antibodies to fight and kill those germs. And not only do proteins help in recognizing germs as part of a glycoprotein, they also help in fighting those germs because antibodies that fight germs are proteins. Proteins can help in transport, moving things in and out of cells. Remember, we said cell membranes are made out of proteins and phospholipids. So if this little pink ring in the animation This little pink ring is a cell membrane. Part of that cell membrane has proteins in it, and some of those proteins are transport proteins. So if my cell needs to stay alive, it needs to take in food, it needs to take in oxygen, it needs to get rid of waste, to get rid of garbage, and there's transport proteins in the cell membrane that help molecules get in and out of cells. And so they're very important in maintaining function, in maintaining cells and keeping them alive. Other ways that proteins help in transport, one of the proteins that's important in transporting in your body is hemoglobin. Hemoglobin is a protein that's found in red blood cells. And its job is to carry oxygen to all your body cells. So when you breathe in and fill your lungs with oxygen, hemoglobin in your blood cells is going to take that oxygen from your lungs and transport it to all your body cells. That's a pretty important function too. Proteins help in moving things around. For example, your body muscles, your muscles in your arms and legs that move your body, are made out of proteins. They also can move single cells. If you look at this little sperm cell swimming, what allows it to move is that big long tail at the end. It's called a flagella. One flagellum, plural flagella. And that little swimming tail that helps the little sperm to get to the egg is made out of proteins. The other way that we move things inside cells, and I know you learned about this in middle school, is mitosis, cell division. When cells divide, remember, we make a copy of our chromosomes and they attach to fibers in the cell. We call them spindle fibers, right? And the fibers that pull the chromosomes, that move the chromosomes apart, are made out of proteins. That mitotic, the spindle we use in mitosis, is made out of proteins. Proteins can also be hormones. Hormones are molecules that are made in one part of your body and used somewhere else. They go around to another place in your body and tell other body parts to do something. For example, when you eat food, your stomach, your digestive tract is going to break down those food molecules and put the glucose into your bloodstream. And there's a hormone that's made by your pancreas called insulin. Insulin is a hormone that's a protein. And the job of insulin is to control your blood sugar. Remember that vocab word from our first slideshow, homeostasis, maintaining a balance between temperature, not too hot, not too cold, not too much sugar, not too little sugar, not too acid, not too much water. Maintaining balance in your body is very important. And insulin is a molecule, a hormone that's a protein that controls your blood sugar. So this is how it works. Pretend this big yellow circle is a cell. Could be a liver cell, muscle cell. And the little red stripe at the top there is a blood vessel going past. And after we eat food, there's glucose in our bloodstream. If insulin is present, what insulin tells the cell to do is take some of that sugar molecule, those sugar molecules that are in your blood, and take them into the cell and save them for later. We don't need to use all that glucose right now. It's a way to maintain the balance of sugar in your bloodstream. And what do we do when we store our sugar? Remember, animal cells take their glucose, glucose. Each one of these little hexagons is a glucose molecule. We store our glucose as glycogen. We store it in our muscles, we store it in our liver. Cells have to take in glucose in order to have the food that they need. And insulin is the molecule that allows cells to do that. People who have diabetes don't make the insulin that they need. So if you don't have insulin, your sugar in your blood, the level of sugar in your bloodstream stays very high. And it's because those cells don't have the insulin to help them and tell them to save their sugar for later. And so people who have diabetes, they have to do a little finger prick. They take a little bit of blood. They're checking their sugar level. If it gets too high, in order to store their sugar into their cells, they need insulin. And if they can't make insulin, what they do is take it as a shot. You can do a shot. Some people have an insulin pump that checks their blood sugar level and gives them the right amount of insulin that they need to keep the balance in their blood. Too much sugar in your blood can cause you to go into a coma, can cause some other serious physical things to happen to you. And so maintaining that balance of sugar in your bloodstream is very important. South Dakota has one of the highest diabetes rates in the nation. And so probably you know somebody in your family or maybe a friend who has diabetes. And maintaining blood sugar is a very important part of that homeostasis. And proteins like insulin help your cells to do that. We have talked about carbs. We have talked about proteins. Our next molecule we're going to talk about is lipids. Stay tuned for the next slideshow to learn about this molecule.